So it's my honor to present the next speaker, Dr. John Lewis. He's a surgeon and man of many other talents, including working in the pharmaceutical industry. He's previously a professor of surgery at Memorial Sloan Kettering, has received numerous awards and accolades, and he's now the CEO of Zyopharm. And he's going to speak to us about systemic therapy for metastatic liver tumors.
Thank you very much. By way of introduction, it's now commonplace for all of us to have one of these. As recently as 10 or 15 years ago, it was unimaginable that there would be so much computing power in something so small at such a low cost. So as we compare this to 45 years ago in terms of NASA putting men on the moon, the computing power that they had was equivalent to about 10% of this. We think that the same or similar is happening and will happen in the world of biology. And I want to give you a brief look into that, highlighting a few examples really in the area of immunotherapy in cancer, because a lot now is coming to age, and how this all could and really should fit together. So by way of disclosure, I am in the biotechnology industry. I'm employed by Zyopharm. I'm partnered with Intraxon. We are both with two companies very focused in this area. I'm a shareholder of both companies. The notion of synthetic biology is not new. In fact, the history, and I'll show you this, goes back about 100 years. A lot of these things kind of 100 years ago, the world started transitioning. But the reality of this is really starting now. So this is from Science in 2011. As always, it's a lot more complex than we first imagined. So these are the equivalent of Lego blocks being put together to make an organism. And while organisms have been synthesized, the complexity obviously is huge. We are not focused on synthesizing organisms, but more on treating disease. And so this is from that same Science article. This is, I think, less applicable to cancer, but it probably will happen in time. But it is applicable currently to several other diseases. And so we and others are currently involved in doing just this. So in other words, reconstituting from a disease state a network that's gone wrong into a network that is quote unquote rewired and is going right. As I say, in cancer, it's less applicable, although in time it probably will be. But for example, in diabetes, okay, where there are sensors for things like glucose, there's ways to secrete glucose and correct a lot of these things. This is something being applied right now. Where we're focused is in the area of synthetic biology, combining engineering and biology. And what's enabled that enormously is the technology revolution. And so the two are really quite intertwined. And so going back again to this device here, which was unimaginable 5, 10 years back, certainly at this cost and this power, it's the same or similar going on right now. And I want to show you a few examples with that. And so what we're doing, and I'll get into some more detail later on, is we have created a tool set where we can, in a really assembly line way, be able to synthesize many different parts of DNA with many effectors, so things like cytokines, antibodies, the short chain fragment of antibodies, et cetera, et cetera. And with that, we can control many cells. We can control them outside the body, ex vivo. Obviously, much more importantly, we can do it in vivo. And I want to give you a few examples of that as we go along here that are currently going on at many institutions. And really, this is an overlap between a lot of government, so many things going on at the NIH and NCI, a lot of industry, a lot of academia, and of course, the investment world is heavily involved in terms of doing this. To go back to the history, and so it was about 100 years ago that this first notion came up, and this, of course, was real kind of fictional science. I mean, this was Jules Verne thinking about the future. It's really accelerated enormously, and so, again, since we put people on the moon, we worked out restriction enzymes, and that was the Nobel Prize in 1978, and that has changed everything we do in biologics. So the ability to make antibodies, therapeutic antibodies, and so on, all of that is predicated on this. Very intertwined with what we're doing now is the start of the information technology revolution, and that was somewhere around about 85, give or take. And I want to go into some of the details there just very briefly in terms of how the cost has come down, because that's a key part of this. And then culminating in, again, the one company I'm affiliated with, Intraxon, who did an IPO, initial public offering, this last August. And this was the largest IPO ever in the history of biotech. And again, just a reflection of the level of interest in this as different people are trying to predict how the future is going to evolve. This is taken from The Economist. This goes up to 2010, but there's two things here. And so if you look at the top line, it's something called Moore's Law. Moore was one of the initial inventors at Intel. And what he predicted is that the cost of storage, the cost of different functions in computers was going to come down, down, down. Again, as he said that, no one believed him, but here it is. At the same time as we sequence DNA, okay, the cost of doing that has come down enormously and even faster as you look at this. And so it really boils down to a lot of the engineering components here, being able to do things much faster, much better, and much cheaper. I want to step back again about 100 years. And so in 1908, Henry Ford had a vision and then executed on his vision. And what his vision was the following, and that is that cars, automobiles had been around for 10, 20, even 30 years at that time. But they were very expensive, so only the very wealthy could afford them. They were very complicated to make. It often took 1 year or 2 years to make one car. And of course, something not broadly applicable. And what he did was he had within his vision a way of doing this, and he applied engineering versus artisans putting these together. And this was the start of the assembly line. And this is an original picture taken again around that time. This was a few years later. But the notion here of having an engineering system to reduce cost, and part of his vision was that what he was building would be affordable to any individual who was earning a salary, period. That was it. And of course, before that time, each car would take years and years to make. Since that time, it now takes a much shorter time. The cost has really stayed the same. The function has continued to improve dramatically. Similar things are going on in terms of the ability to apply synthetic DNA and synthetic cell systems. And so part of that is having an assembly line. We can manufacture synthetically hundreds of thousands or millions of components of DNA. And we can put those together, and we can do it quite well with DNA robotics. In addition, because of the computing power available now, and all of us use the cloud to whatever extent, whether it's Amazon, Dropbox, whatever, and these are server farms with enormous computing power. And it's this combination together that's enabling an engineering type approach to developing new treatments in cancer. This is NCI and NIH data, and again, just verifying and showing, this goes to 2013. Moore's Law continues, and so the cost, again, of computing power keeps on coming down. At the same time, the cost here of looking at genome, in other words, getting a sequence report, has come way down, it continues to go down. And at the same time, the cost of synthesizing, not shown here, is coming way down. This is all great, but it's, as always, it's never easy. And so there's several components of this that are very complex, very undefined. This complexity is unwieldy. A lot of these parts are incompatible. And so again, it's not taking Lego blocks and putting them together, but it's an enormous amount of learning and understanding. So there have been tremendous challenges. Some of those are being overcome, some are not. With that, what I want to do, and perhaps in this context, is show a short 2-minute video. This is a video, and it's an advertisement from an investment fund. This is a large institutional investor called Fidelity, actually in part based out of Rhode Island, in part based out of Massachusetts. Again, by way of disclosure, they are investors in terms of the two companies I mentioned before. But with that, let me go ahead, understanding all this complexity, and show their video.
There are 1.7 million known species of life on Earth. 2 years ago, scientists introduced the first one ever designed by a computer. And in the last 10 years, there have been over 3,000 patents issued for genetically modified organisms and other transgenic innovations. Within 50 years, we could have more life forms invented in the lab than we've ever identified in nature. We now have goats whose milk can be spun into spider silk that's stronger than steel. Jumbo salmon that grow twice as fast as their natural cousins. Bacteria that produce anti-malarial drugs once available only from plants. Innovations like these can increase the supply of essential products, inspire new investing ideas, and launch or upend entire industries. It's all part of a new science called synthetic biology, using nature as a manufacturing platform and DNA as the raw material. Pharmaceutical companies see it as a pipeline for extraordinary new drugs and treatments. Energy companies see a route to cleaner, more sustainable fuels, like algae that produce biofuels and eat carbon dioxide. Someday, computers may run on DNA-based circuits, and biological paint could help heat and cool your home. Around the world, across borders, academics, entrepreneurs, and even students are working with over 5,000 DNA sequences called biobricks to explore ideas and invent new organisms. The DNA is available online in an open source database, and the collaborative, crowdsourced approach means experiments that used to take years now take weeks, constantly redefining what's possible. Although synthetic biology is still in a very early experimental phase, it could become the defining technology of the 21st century, bringing with it radical new thinking, new questions, and new opportunities. Because nothing has the power to change how we live more than changing life itself. Think about it. We do.
So I think, and again, as we're really just beginning to start this, but this could well become the defining technology of this century in many respects. What's critical is to be able to apply it in ways that are going to be sustainable and meaningful, in particular as we look at human diseases. And so what I want to do now, again, is focus on cancer and focus specifically on the area of immunotherapy. Again, the core principle of synthetic biology is to have all these different tools, and there's a lot of them, and to be able to do it faster, better, cheaper, FBC, to be able to, like engineers do with cars, design, test, build, and load. And if we go back to the Henry Ford story, it's about 100 years ago that this whole thing started. The rate of improvement keeps on getting better and better every year. It's astonishing. One measurement of that is the number of deaths, highway deaths in the United States have come way down because cars are getting safer and safer. Yet the cost, as we look at that, if it's inflation adjusted, has stayed relatively constant. And a lot of this goes back to, again, the Ford vision and the Ford implementation of engineering principles. So what we've done is, and this has gone on within Intraxon over the last 5 years, is built an enormous library, an enormous tool set that again is it's not quite on the shelf as one would have in terms of some kind of manufacturing system, but it's stored in different parts that a DNA robot can easily access or a cell robot can access. So stored under the correct conditions for DNA and cells. The effectors we're looking at in cancer include cytokines, and we're doing one of those studies now, monoclonal antibodies, short-chain fragments of monoclonals, and I'll get into some of those details, and then a variety of others, protein ligands, intracellular decoys, and of course RNA. The cells that we are involved with include many of the immune cells, so T cells, and I'm going to focus down on that shortly, all the other ones, dendritic cells, etc., and then a whole host of non-immune cells, and that of course includes the tumor cell itself, something called MSC, mesenchymal stem cells, and I think we will be hearing a lot more of that in time. These are allogeneic cells that are engineered with different components of DNA, and what they do when they're injected is they track to the site of a tumor in vivo. And then of course all the other cells, including epithelial, endothelial, etc. And obviously there's many ways to get this to function. But again, what I want to do is just focus down now on how this is being applied, and I think will be applied to cancer immunotherapy. And of course, cancer immunotherapy has evolved enormously over the last few years. There have been really two broad, huge discoveries that have moved the needle enormously. So one is checkpoint inhibitors, and that's in particular in melanoma, but many other cancers now as well. But that clearly has evolved enormously. And the other is cell-based therapy. There's a rationale for this, and of course we've been working in immunotherapy in many ways now for years, but a lot of it is becoming clearer, some predictable, some not predictable. But part of this rationale is the following. And so what has become quite clear in breast cancer, as an example, is when we compare DCIS, so something in situ, not cancer, something that may become cancer, often does, to invasive ductal, and we compare the actual cells themselves, so the cells of DCIS to the cells of an invasive ductal carcinoma, the genetics of those cells are often very similar. In fact, it's remarkable how more similar they are than different. One of the key differences that is emerging is the following, and that is that the surrounding cells are very different. And these are cells derived from the bone marrow and the immune system. And so one of the key differentiators that's emerging right now between DCIS and breast cancer is that DCIS has has many tumor-infiltrating lymphocytes, okay, that are relevant in terms of an anti-cancer response, whereas the invasive ductal has much fewer of those and many aberrant immune cells. In fact, often they look like leukemic cells, okay, that are next to breast cancer cells. This is not just a picture, because as we go through data, and so one of the key presentations at the San Antonio Breast Symposium this last year was that as we look at the two different treatments, one is giving chemotherapy to women with breast cancer, the other is using antibodies, so Herceptin. One of the biggest predictors of prognosis, in other words, one of the biggest predictors whether that woman is going to live or die, is the number and type of bone marrow derived and T cells that are in the tumor. And those with better and more T cells do much better. They live. Those without those often don't live. So with that as a background, there's been a lot of work that has gone on. And this is taken from the NCI, from the surgery branch at the NCI. And what Rosenberg and his team have really evolved enormously, especially over the last few years, is the idea here of an adaptive immunotherapy. In other words, getting TILs, tumor-infiltrating lymphocytes, from the tumor, expanding them ex vivo, then coming back and changing the compartment of the bone marrow using things like chemotherapy and replacing it with these cells. And certainly in melanoma, and probably other solid tumors as well, it's been astonishing how well this has worked. What's gone on at the same time, and this is many places doing this, again, I'm going to refer to NCI data, but this is going on at many academic institutions, so Sloan Kettering, UPenn, a series of others in Texas, and now many different companies that are forming around this, is the idea of taking CAR, chimeric antigen receptor, okay, on a T cell and using that as immunotherapy. And the data have been astonishing, truly astonishing. And so what's done here is that a T cell is genetically engineered. And this is, again, this is an example of synthetic biology to some extent. A lot of this now is being done in an artisanal way, so it's artisans doing it, individuals doing it. But what's being done is connecting on the outside a short chain fragment of an antibody that will bind to a defined cancer cell, and then on the inside, different components that will activate their T cell. And it has been astonishing how these activated T cells can reduce cancer. So far, most of this has been in the setting of liquid tumors, so things like lymphoma, leukemia. It has been much more difficult in solid tumors, but certainly I and we believe that that is going to happen. And with that, I want to show you some of the data. And so these are data presented recently. This is ASH, the American Society of Hematology, this last year, December. These are data taken from the NCI, and the target here, the short chain antibody fragment, is going after CD19 cells. And this is in the setting of lymphoma. And this picture tells 1,000 words. These are extremely, extraordinarily, heavily pre-treated patients who have been refractory to anything known. Okay? All of them have been on a course to die. And again, the response rate that's seen here is extremely high. This has been replicated by others, so again, at Sloan Kettering, at UPenn, various other sites and centers as well. What's notable here is that, and these are from the data, the acute toxicities here are downplayed to some extent, but we know very clearly that it's really not the case. So the toxicity can be severe, and it often kills people. And to that extent, recently, a week or two back, Sloan Kettering, doing a similar study, have been put on clinical hold by the FDA because of 2 patient-related deaths. This is all fixable and it's all going to happen, but the point here is that this is not trivial. Some of the side effects, which is really an immune-activated multi-system organ failure, can be severe, but there's several ways of dealing with it. Again, many are forging on now into solid tumors, and so this is a study that just started a week or two back at the NCI. This is the pediatric branch at the NCI. And this is a phase 1 study in pediatric osteosarcomas and sarcoma patients. And this, again, will go into full play within the next few weeks, next few months. And part of what they have is a way to kill these cells in the event that these side effects happen. What I'm going to show you now is some unrelated data, okay, in terms of some of the systems we are working with, where in vivo we can turn genes on and off and then on again and on and off as many times as we need to. This is an in vitro system showing cells in culture. What we're doing here is taking a gene that is ordinarily in the off position, and through a pill that's taken, it goes into the on position. These are data taken from human subjects with melanoma and breast cancer, and what this points out is that we can go from off to on to off and then back on again by either giving or not giving a pill to people. We can also very tightly control the dose. And so through things like this that we believe the future of something like CRT therapy is going to evolve, and evolve in a more rational way. And so as we go from second to third to fourth generation, components are going to be built in here that will enable very tight control. So as soon as things start going wrong, and when they go wrong, it happens very quickly, the same as acute multi-system organ failure in a septic patient. There's ways to control that, and control it where you can turn it off and on again. And that's just a brief insight. As important is the following, and that is that I don't know what the exact cost of current CRT therapy is. The NCI, again, are so heavily funded that they don't really know what the cost is. I believe at a place like Sloan Kettering or UPenn, it's somewhere around about several $100,000 per patient, so probably somewhere around about $500,000 per patient. And to go back to, again, the vision of Ford, and it's not just Ford, but this is a committee that ASCO has put forward over, again, the last few weeks. So the public announcement came last week, but they've been working on this the last few months, weeks, and again, this question that comes up over and over in terms of value, and a key component of value, as Ford articulated, is that everyone should be able to afford it. Any person earning a salary should be able to afford it. So cost is absolutely key. And it's certainly our belief, our vision, that using these engineering approaches, we'll be able to apply this to several different treatments in cancer and reduce that cost. And again, it's coming off, and in fact, a real mirror image of what's going on in technology. Again, the ability to hold this phone that has such enormous computing power, yet is affordable, because I think everyone in this room is holding one. As importantly, millions of people outside of this room, again, earning a normal salary, can hold this phone. And so in conclusion, as we look at the near future, what is going to happen is the following, and that is that engineering principles are being applied and will be applied to product development in cancer. As we do that, and it's again by designing, building, testing, and learning, as we do that, different components are going to be much more scalable, again, very much like the Model T Ford, So suddenly we can do a lot more at a much lower cost. So the COGS, the cost of goods, comes way down. And we and many others believe that this is a near-term reality. It's going to happen more and more in terms of many treatments that we do in cancer. And again, to conclude, it's this algorithm that we've done so well with technology, and we're beginning to do now, and we will do better and better as we develop better products in cancer, of being able to do it faster, better, and cheaper. With that, I will conclude, and I want to take this opportunity to thank the organizers of this for including me and for organizing such a great conference. Thank you very much.